Okay, good morning. Okay, so today let's finish the carbohydrate metabolism, the pathways, gluconeogenesis and pentophosphate pathways, right? And then we have a quiz, right? And we already talked a little bit about the gluconeogenesis. Gluconeogenesis definition definition is the conversion of non-sugar precursors to glucose, right? I mean the body that utilize non-sugar precursors to make glucose. And which physiological conditions that gluconeogenesis would be activated? Fasting, right? So during fasting, our body, there's a lot of gluconeogenesis. The main purpose of gluconeogenesis is to, what is the main purpose, or what is the function, why we need to do gluconeogenesis during the fasting? To raise blood glucose level, right? To not allow the glucose level in the circulation too low. And this is very important because some of the organs, tissues, heavily rely on the glucose for energy, like red blood cells, almost 100% rely on the glucose for energy, right? Because red blood cell cannot use other energy fuels. And also brain is very important, right? And most of the energy that for brain is actually from the glucose. So, and we also talk about the pathways. Remember that I always ask you guys to remember the key reactions in each metabolic pathways. In the glycolytic pathway, we have three very important so-called rate-limiting reactions, right? And these three rate-limiting reactions cannot be reversed. It means that this key enzyme in the glycolytic pathways only catalyze the reaction go this direction. I cannot go the opposite direction, right? So that means our body needs a different set of enzymes to catalyze the reaction towards gluconeogenesis, right? So, and I also talk about the three bypass reactions in the gluconeogenesis pathways. And the first one is like, converted pyruvate to phosphoenolpyruvate, and this reversal pathway or bypass pathway requires two enzymes, right? Pyruvate carboxylase and phosphoenolpyruvate carboxylase. And the second bypass reactions, drive the reaction to the generation of fructose 6-phosphate, and this reaction catalyzed by key enzymes called fructose 1,6-biphosphatase. And the last one, which is very important one, that is the conversion of glucose 6-phosphate to free glucose. And this reaction catalyzed by glucose 6-phosphatase. Remember, the muscle lacks this enzyme, right? So muscle is not able to make or directly release glucose into the circulation. And this, I just give you an overview of the gluconeogenic pathway, and just you try to focusing on these key reactions, right, in the gluconeogenesis. And two reactions that occur in the mitochondria, and the other two reactions in the cytosol, right? And also I mentioned that depend on which precursor that our body use, and then the gluconeogenic pathway could be different, right? For example, if we use a lactate or pyruvate as the precursors, then it involves all 4 reactions, right? 2 in the mitochondria and 2 in the cytosol. And if glutamate as the precursors, then we only utilize 1 reactions in the mitochondria and the 2 reactions in the cytosol. And glycerol, if we use the glycerol as the precursors and we only 2 reactions, right, undergoing the 2 reactions in the cytosol. So now let's go to a little bit detail that in the mitochondrial reactions, the 2 reactions, right, catalyzed by pyruvate carboxylase and PEP-PCK or phosphoenolpyruvate carboxylase. Again, if lactate or pyruvate as the precursors, and it actually goes to 2 reactions, right? In the mitochondria, so involve 2 reactions. And the PEP, phosphoenolpyruvate, is the product that's generated in the mitochondria and then transported into the cytosol and finish the rest of the reactions in the cytosol, right? And oh, I forgot the other one. So glutamate, right? It's glutamate is the precursors, and glutamate will directly convert it to oxaloacetate and PEP, and do not involve, and this pathway does not involve the pyruvate carboxylase reaction, right? So you know the glutamate is converted to alpha ketoglutarate, and alpha ketoglutarate enter the TCA cycles. Remember, I also mentioned the TCA cycle involved in the gluconeogenesis. So alpha ketoglutarate enter TCA cycles and goes several reactions and then converted to oxaloacetate. So oxaloacetate then leaves the TCA cycles and be converted to PEP, right? So glutamate as a precursor only involves 1 reactions, right, which is catalyzed by PEPCK enzyme, right? And glycerol only involves the 2 reactions in the cytosol. I can, short pathway, right? So first of all, glycerol can be phosphorylated to glycerol 3-phosphate by the enzyme called glycerol kinase, right? So glycerol kinase is only present in the liver, right? So that's why liver is able to utilize glycerol to make glucose, right? So this is very important. Glycerol kinase is a very important enzyme for gluconeogenesis from glycerol in the liver. And then glycerol 3-phosphate can be converted to these molecules, right? And then enter the rest of the pathways of gluconeogenesis converted to glucose, right? It involves these 2 key enzymes in the gluconeogenic pathway. Okay, so these are the 2 last reactions in the cytosol that converted the intermediate to eventually to the glucose, right? So I highlight here is the blue. This represents the glycolytic pathway, right? And the red is the gluconeogenic pathways. So let's look at the first bypass reactions. This is it's involved the enzyme called fructose 1,6 biphosphatase in the gluconeogenesis, and what this enzyme does is to convert the fructose 1,6 biphosphate to fructose 6-phosphate, right? So this is again the key enzyme in the gluconeogenic pathways. And just the in parallel, the phospho fructose 1 kinase is the key enzyme in the glycolytic pathway, right? And catalyze the opposite, reaction. And the second reaction or the last reaction in the cytosol in the gluconeogenesis is the enzyme that involves the enzyme called glucose 6-phosphatase, right? Again, this is the last reaction in gluconeogenesis, and this enzyme converts the glucose 6-phosphate to glucose. So remove the phosphate group from the glucose and release the free glucose, right? So this enzyme again is very important, the key enzyme that determines the whether this tissue be able to release glucose, right? Okay, now let's talk about the specific cycles or pathways for gluconeogenesis. The 2 pathway of cycles that you guys probably wanted to remember. First one is the Cori cycle. So Cori cycle basically is the gluconeogenic pathway that utilizes lactate as a precursor, right? So lactate is mainly from muscle and the red blood cells. The red blood cells, because it utilizes glucose, right, and cannot complete oxidize. So red blood cells actually generate a lot of lactate and release into circulation. And muscle is also release a lot of lactate, particularly during the fasting. A lot of tissue try to spare glucose, not to completely break down glucose. Always, keep the or save like carbon skeleton. So under fasting conditions, muscle also does the partial oxidation glucose and release a lot of lactate. So lactate concentration will be very high in fasting conditions, and that provides precursors for gluconeogenesis in the liver. So liver picks up a lot of fatty lactate from circulation and use this as a precursor to make glucose in the liver, right? So this cycle is called a Cori cycle, meaning that lactate produced from red blood cells and skeletal muscle and utilized by liver to make glucose, right? So this is a cycle. And once it's glucose made in the liver and then released back to the circulation to raise the blood glucose level and also provide glucose for energy utilized by other tissues, particularly in the red blood cells and brain, right? During the fasting condition. And the second cycles are called alanine cycles. So alanine cycles that involve actually only 2 tissues, muscle and the liver, because alanine, again, during the fasting condition, and amino acids can also be utilized for the energy. The protein can be break down and then utilized as energy. So muscle releases alanines and provide alanine for gluconeogenesis in the liver. Alanine is also the precursors of gluconeogenesis, right? And the reaction, for example, amino acids, other type of amino acids can be metabolized to produce a pyruvate, and then pyruvate being converted to alanine through called a transamination reaction, and release this into the circulation and pick up by the liver. So in the liver, alanine being converted back to pyruvate and enter the gluconeogenic pathway for generating glucose, right? And then glucose released into circulation. So this cycles is important for the gluconeogenesis, right? Any questions? Yes. Yeah. So that's why this is called alanine cycles. And I just want to mention, because in the liver, alanine is converted to pyruvate, right? Pyruvate then enters gluconeogenesis. So if alanine as a precursor, then it will also go to the 4 reactions, right? Okay. Here I just give you the details for the gluconeogenesis. Glutamate, if glutamate as the precursors, again, it also only involves 1 reactions, right, in the mitochondria, and for generating PEP and PEP. So, and its important thing is that this precursors, glutamate as precursors that involve the TCA cycles, right? So TCA cycles involved for gluconeogenesis when we use the glutamate as a precursor. And here's the TCA cycle again. I just wanted you to know how glutamate, how TCA cycle involve the gluconeogenesis. So for example, glutamate, right, first of all, it converted to alpha ketoglutarate and enter the TCA cycles, right? And through the several reactions, then alpha ketoglutarate can be converted to oxaloacetate. So oxaloacetate being converted to PEP, remember, right? And lead the TCA cycles, enter the gluconeogenic pathway for glucose production. So this just gives you some idea, and also the other amino acids that if they can be converted to succinyl-CoA, and a similar thing that this amino acid can also involve TCA cycle for gluconeogenesis, right? Just give you kind of, overview or idea how TCA cycle is involved in the gluconeogenesis. So in other words, that basically when amino acid as a precursor, that TCA cycle is involved, right, for gluconeogenesis. And other thing is, since we're talking about TCA cycle here, I also want to mention that the acetyl-CoA, right, remember, is a key central molecule that being generated from the oxidation of amino acids, glucose, and fatty acids. And this is the molecule that first enter the TCA cycles, right, for oxidation. And also citrate is a key molecule in the TCA cycle because it can be used for fatty acids synthesis. So this is also the key molecule that connects carbohydrate metabolism to fatty acids synthesis or metabolism, right? Or again, I mean, citrate not just the carbohydrate but also amino acids. Connect the amino acids, carbohydrate to fatty acids metabolism. We will talk about later that not just the excess carbohydrate intake can be converted to carbohydrate can be converted to fatty acids, but amino acids can also be converted to fatty acids, fat, right? If we consume too much, right? And here is the process for the gluconeogenesis. from the glycerol, right? And again, this occurs only in the cytosol, does not involve mitochondria, and only 2 reactions in the cytosol, right? And again, glycerol kinase is very highly active in the liver, so the gluconeogenesis, I mean, if glycerol is used for gluconeogenesis, that only occurs in the liver because of this high activity of this enzyme, right? Okay, so energy utilized in the gluconeogenesis, the whole process it requires 6 ATPs, utilize 6 ATPs for gluconeogenesis, and if pyruvate is utilized for the gluconeogenesis, right? And the energies of this ATPs and mainly from fatty acid oxidation and partial amino acid oxidation, right? Okay, here is the slide, and I guess just give you big pictures of the gluconeogenesis, and you can see the 2 cycles also that involving is present in this slide, right? So again, the alanine cycles again only involve in the alanine, but actually if we extend a little bit, so I think not just the alanine, and maybe your question is I wanted to revisit your question. So not. I mean, alanine is only the alanine that can be picked up by the liver, but alanine can be from the other amino acids, right? So other amino acids, any type of amino acid that can be converted to pyruvate in the muscle, that can be used for generation of alanine. Right? So in other words, the alanine cycle not just only involved in the alanine, but also other type of amino acid that can be converted to pyruvate alanine. Right? So this alanine cycles really, I would consider this as the gluconeogenic pathways, and these pathways that utilize the amino acids as precursors. Right? Does that make sense? Okay. And the Cori cycles basically utilize the lactate as the precursors, right? So lactate from released from muscle and also red blood cells as well. And then pick up by all these precursors can be picked up by the liver and form glucose, right, in the liver. So again, the glucose, why we need to maintain the blood glucose level and during fasting, and because the brain and the red blood cells and other type cells that will heavily rely on glucose for energy, that requires, we have the high levels of glucose in the circulation. Now let's do some, I guess, interesting thing. The clinical connections, right? Remember that PCK is a key enzyme in the gluconeogenesis pathway, right? What happened to the blood glucose level, and blood glucose control, and other the important intermediates in glucose metabolism in a patient if they don't have a PepCK. That's a key enzyme in the gluconeogenic pathway, right? First question, what happens to, excuse me, blood glucose control, blood glucose level, particularly during the fasting? If they don't have the PepCK, the key enzyme, Yes. Yes, remain low, right? They're not able to remain the high blood glucose level, so will go so-called hypoglycemia. They have a lower glucose level, blood glucose level. And what happened to the blood lactate levels compared to the normal person? Yeah, high, right? So lactate level will be very high because this is so-called a lactic acidosis. Because when our body blood glucose goes low, and we always try to spare glucose, so not a lot of tissue will try to spare glucose by partial oxidizing glucose, right? So a lot of tissues only that do partial glucose oxidation. That means that when glucose will not be complete oxidized, just stop at pyruvate and lactate, right? So then a lot of tissue will produce a lot of lactate and release into circulation. So this is the reason. And what happens to the glycogen store? If we can make a glucose from non-glucose, yes. Stay high or low? Why you think it's high? You know, what we store glycogen for? Make glucose, right? And because we cannot make glucose from gluconeogenesis, then we have to use more glucose from glycogen breakdown, right? Right? So then the glycogen level should be lower. Does that make sense? Right? It's called glycogen depletion. Remember, during the fasting, we have two pathways that can contribute the blood glucose, right? One is to, actually, glycogen is the first, glycogen breakdown is the first pathway that can be activated and to provide a glucose into the circulation. So, and the second pathway is then, when we don't have second pathway is to make a glucose, right? Yes, the question.
In a fast state, would your glycogen store still be able to be a normal level just because you have a defect in the enzyme? While you're eating, would you still be able to make those glycogen stores?
Good question. I think so, yes. But maybe compared to I think that the enzyme itself may excuse me, may not affect the glycogen store, when you eat, right? I mean, you have a lot of glucose, so we make glycogen to store. But when you fasting, then you need more glucose, and not only from, glucose during the fasting from the two sources, right? Glycogen, break down the glycogen, and the gluconeogenesis. So if one source has a problem, then the other source we have to overuse, glycogen. So you break down a lot of glycogen, try to maintain blood glucose level. So in this patient, if they don't have a PEP-CK enzyme that they can make glucose from non-sugar precursors, and they overuse the glycogen, right? Does that make sense? Okay. And what about the other key enzyme, the glucose 6-phosphatase deficiency? What happened to the blood glucose level? Yes. Yeah, it would be low, right? Hyperglycemia, the same reason. And this is also the the key enzyme for gluconeogenesis. If this enzyme is defect, then glucose cannot be made, right? And particularly it's also happening during the fasting. What happened to the lactate levels? Similar thing, right? Similarly, you also have a higher lactate level in the circulation. An interesting thing here is what happens to glycogen store in this patient with glucose 6-phosphatase deficiency. Yes. Right. Right. This is the enzyme. Say that again. Sorry. That's a good question. So what do you think? The glycogen store would be lower or? Correct. Yeah. So that means that glycogen will be accumulated, right? So it's different from the deficient in the PepCK and deficient the glucose 6-phosphatase deficiency actually causes glycogen accumulation. So this patient, I think the hypoglycemia even more severe compared to PepCK, right? This patient cannot utilize glycogen, cannot break down glycogen to release glucose, right? So actually both pathways having problem, right? Cannot break glycogen to release glucose and also cannot make gluconeogenesis. So the reason is here, and this is the enzyme, right? This is a reaction that this enzyme catalyzes, right? Catalyze the conversion glucose 6-phosphate to glucose, right, in the gluconeogenic pathway, and also glycogen breakdown pathway, remember, right? So glycogen breakdown, glycogen eventually break down to the glucose 6-phosphate and require this enzyme to release free glucose. Yes. Yes, right. The same enzyme that's missing in the muscle, so that's why the reason this cannot become, converted glucose, free glucose. So you have a lot of glucose 6-phosphatase accumulated in the liver, and this glucose 6-phosphate is very strong inhibitor of the glycogen breakdown pathway, right? We're going to talk about later when we talk about the regulation of metabolism. So the reason why you have a lot of glycogen accumulated because this is the key substrate that accumulated in the glucose 6-phosphate deficient liver, and then it's inhibit or block the glycogen breakdown pathway. So glycogen cannot be break down even, right, to release glucose. Okay, now let's talk about gluconeogenesis in the kidney. It occurs during the prolonged starvation. So again, the liver is the most important tissue that produces glucose, right? Does the gluconeogenesis, particularly during the early stage of starvation. When starvation is prolonged, for example, takes longer than 24 hours, and then kidney then also become important sources of glucose, right? So kidney also does the gluconeogenesis, but it's a little bit late, compared to the liver. And for example, at the very later stage, and kidney also become important, like contribute 50% of glucose synthesis to the circulation. And it's also different from the gluconeogenesis in the liver. The precursors that are utilized for the gluconeogenesis in the kidney are mostly amino acids, right? So gluconeogenesis is the kidney, and you basically utilize the amino acids as a precursors. And the pathways, again, I mentioned before, the in gluconeogenic pathways, and that if we utilize amino acids as the precursors, always involve TCA cycles, right? So called an anaeroprotic and catalytic reactions. This we will talk about the next couple of slides, right? This process reactions that involve in the whole TCA cycles, right? Okay, now anaeroprosis and catalysis. So these are the enzymatic reactions or pathways that replenish the pool of the metabolic intermediates in the TCA cycles, right? So this two are the different process that is this is the purpose of our body. Does the anaeroprosis or catalysis is to replenish the pool of intermediates in the TCA cycles, and try to keep the pool size of TCA cycle intermediates constantly, right? Not to allow the too much intermediate accumulate or depletion of intermediates in the TCA cycle. And the purpose of this is to maintain the normal function of TCA cycles. And these two processes has a different definitions. The anaeroprosis means that the adding the intermediates into the cycles, right? To not allow the depletion of the intermediates in the TCA cycles. And catalysis means that some of the intermediate can leave the TCA cycles and go to the different pathways. So again, the anaeroprosis and catalysis need to be balanced, and purpose of this process is to maintain the normal function of the TCA cycles, right? To keep TCA cycle running. And more information about these two processes. For example, and this reactions that we actually adding the alpha ketoglutarate, which is the intermediate of the TCA cycles, right, to the TCA cycles. So this process reactions we call anaeroprosis, right? You adding the and similar for these reactions. And just wanted to finish all the anaeroprosis reactions. Okay, so all these reactions that you can see, the adding, the intermediates TCA cycle to the TCA cycles, right? So all this process are called anaeroprosis or anaeroprosic reactions, right? And catalysis, this two that highlight red is considered catalysis, right? Because you see the citrate leave the TCA cycles and it converted to fatty acids. And oxaloacetate also leaves the TCA cycles and enter the gluconeogenesis pathways, right? So, for example, during the gluconeogenesis, during the fasting, when gluconeogenesis pathways are very active, so you have a lot of oxaloacetate being used for gluconeogenesis, right? So you need to replenish the oxaloacetate. So lots of amino acids that can be utilized, right, for gluconeogenesis and converted, for example. The glutamate being added, convert, the alpha ketoglutarate, and this can be converted to oxaloacetate to replenish oxaloacetate, right? So this will keep, TCA cycle running, and be able to make glucose during the fasting conditions. And this catalytic process, the anaerobic process, also involve the other metabolic reactions or pathways. For example, here we, I think I already described this, right, process, gluconeogenesis. And you add in the alpha ketoglutarate from glutamate and to make oxaloacetate for gluconeogenesis, right? So this will be considered anaerobic process and this catalytic process, right? So catalytic process, anaerobic process are involved in the gluconeogenesis in the TC cycles. And this process is also involved in the glutamine metabolism in small intestine. Remember, the small intestine utilize a lot of amino acids for energy. So glutamine is one of the amino acids that can be utilized in the small intestine for energy. So you can see how glutamine being metabolized for energy production. Interestingly, glutamine, first of all, be converted glutamate and alpha ketoglutarate enter the TC cycles, and alpha ketoglutarate eventually being converted to malate, and malate leave the TC cycles, which is called a catalytic process, right? And malate being converted to oxaloacetate and to form the PEP, and PEP being converted to pyruvate, and pyruvate eventually being oxidized to acetyl-CoA and enter the TC cycle for oxidation. So it takes the long, pathways to be oxidized. So, this is just what I probably did. And this is the called a glycerol neogenesis. How glycerol is being made from amino acids and glucose. Remember, mentioned that carbohydrate, glucose, or or amino acids can be used for fat formation. I'm sorry. Try to finish it. Maybe just take a break. Sorry. All right. Okay. So let's finish. Um, so this slide shows the glycerol neogenesis, means that the cells of our body is able to utilize the amino acids or glucose to make glycerol backbones for triglyceride synthesis, right? So this whole process, how amino acids and glucose can be converted to glycerol phosphate, which is the backbone, remember, for the triglyceride synthesis, right? And first of all, both the amino acids and glucose can be metabolized. First of all, the pyruvate, and then transported to mitochondria. And this is an important pathway, right? So some of the pyruvate can be directly converted to oxaloacetate, enter the TC cycles, right? And oxaloacetate being converted to malate and leaves the TC cycles and convert into the cytosol and convert it back to oxaloacetate and PEP, and then to the glycerol 3-phosphate, the backbone of the triglyceride, right? Yes? Oh, what physiological conditions? Yeah, it should be in the fat condition when we have, consume a lot of nutrients, or a lot of amino acids and carbohydrates under these conditions, right? Okay, so now this is the, we finished 2 pathways, right? Glycolysis, TC cycle complete oxidation of glucose, and gluconeogenesis, right? The 2 pathways we already finished. And then this is the 3rd pathway of a glucometabolism, the pentose phosphate pathways. So pentose phosphate pathway, the purpose of this pathway is to provide the 5 carbon sugars and the nucleotides, right? So, and the first or starting molecules of this pathway is glucose 6-phosphate. So glucose 6-phosphate can enter the pentose phosphate pathways for generation of 5 carbon sugars, and the basically for 5 sugar powder, and 5 carbon sugars can be used for synthesis of nucleotides like DNA, RNA, NAD, or NADH stuff, right? So this is just the pathway. I don't think you need to remember exactly each reactions, enzymes, right? Just try to remember the substrate or the first starting molecules of this pathway is glucose 6-phosphate, right? And what is the purpose of this pathway, right? For the generation, again, the 5 carbon sugars. And it also generates NADPH, right? It's not NADH, it's NADPH. So NADPH is very important for fatty acid synthesis and other important function we're going to talk about a couple of slides later, right? So this, I think, is all you need to remember for these pathways. So this is the so-called secondary pathway glucooxidation, the sources of, again, I mentioned this is pathway, one of the important function of this pathway is to produce NADPH, right? And also the production of 5 carbon sugar phosphates, like ribosome, ribose 5-phosphate, and cells 5-phosphate, right? And this 5 carbon sugar phosphate a very important precursor for nucleotide synthesis, right? And this reaction takes place in the cytosol. Here, more information about NADPH. So NADPH is required for the synthesis of fatty acids, cholesterol, or sterols. Right? A lot of tissues, be able to utilize, be able to synthesize fatty acids or cholesterol, or sterols, right? And it requires NADH for protons or electrons, right? And for reaction, for oxidation reduction reactions in this process. And NADH is also important for maintaining the integrity or membrane integrity of red blood cells. So this pathway is also very active in the red blood cells, right, to produce NADPH, which is important for the membrane integrity, right, in the red blood cells. And ribose 5-phosphate is essential, right, for the biosynthesis of nucleotides and nucleic acids like ATP, coenzyme A, NAD, NADP, or FAD, RNA, DNA, all this kind of stuff actually is synthesized from ribose 5-phosphate, right? So again, this pathway is relatively simple, but just try to remember the function of this pathway is to provide NADPH and 5-carbon sugar phosphate, right? And 5-carbon sugar phosphate is very important precursor for the nucleotide and nucleic acid synthesis, right? Now let's talk about the regulation of glycolysis and gluconeogenesis. So we now understand the two main pathways, right, in the glucose metabolism, glycolysis versus gluconeogenesis. And these two pathways is active in the different conditions, right? The glycolysis being very active in the fat conditions, and gluconeogenesis basically is more active in the fasting conditions. And our body, need to maintain glucose, need to activate glycolysis or gluconeogenesis, again, depend on the physiological conditions. So how our body knows, when to activate gluconeogenesis, or something like this. This is something we're going to talk about, is regulation, right? So regulation of these two important metabolic pathways can be accomplished by the three main aspects. The first, the glucose transport. Remember, when glucose enters the cells or tissues, that are regulated by glucose transporter, right? Different tissues have different transporters, and they regulate differently. For example, GLUT2 in liver and pancreas, and basically it's more active when blood glucose levels high, right? So basically regulated by glucose levels, and GLUT4 mainly in skeletal muscle and adipose tissue, and more active or are more regulated by insulin. So transporter is at least is one level of a regulation, right? It can regulate the glucose metabolism or glycolysis, for example. And substrate cycle. Substrate, during the metabolism of either gluconeogenesis or glycolysis, and produces some important intermediate substrates. And these intermediate substrates are very important regulators of the key enzymes, right? So then can control the whole pathway, the activities or rate. And lastly, hormonal regulation. We already learned that insulin definitely is the most important hormone that be able to regulate enzyme activities in each pathways, right? And we're also going to talk about other important hormones like glucagon, right? Glucagon is a hormone that increase during the fasting, right? And can regulate, the gluconeogenic enzymes. And transporters, and here is a table. I list again the 5 different transporters that we talk about it. And 2 more things that I added to the table is the affinity for glucose and the Km. Affinity for glucose, that means how easy that the glucose be able to bind the transporters and can be uptake, by the transporters. So if these transporters has higher affinity for glucose, mean that this transporter is very easy to the glucose is very easy to bind these transporters, right? So then be take up. And Km is just the opposite to the affinity for glucose. If glucose transporter has a high affinity for glucose, but usually have a lower Km. We're going to explain later why affinity and Km is opposite, right? So here I just wanted to introduce the Km to my Michaelis constant Km. So this Km usually is defined as the substrate concentration at half the maximum velocity, right? So do you guys learn about this like Km before? Okay. So this is how Km is defined. Usually when let's see, if we keep the amount of enzyme concentration the same, and then if we adding the substrate, so if we adding more substrate, then reaction will be increased, right? If enzyme concentration keep the same, and if we adding more substrate to the reaction, then the reaction speed will speed up, right? So this will sort of have a linear relationship with the substrate concentration, right? So, but once you add enough high enough concentration of substrate, then reaction will not increase anymore. So reach the maximum reactions, right? So when you reach the half of the maximum reactions, how much substrate is needed for reach the half the speed of the reaction. So this substrate concentration is defined as Km, right? So this is something also related to the affinity. For example, if enzyme has a higher affinity for the substrate, then you don't need adding a lot of substrate to reach the half maximum reaction, right? The speed of reaction. So that's why the Km is opposite to affinity. So if enzyme have a high affinity for substrate, and it should have a lower Km. Does that make sense? Right? Okay, so now let's talk about the glucose transporters. About the Km affinity for glucose. Different glucose transporters have different affinity and Km for glucose. For example, GLU1 and GLU3 basically have a low Km or high affinity for glucose. So that means that only when blood glucose level reach very high, then these two transporters can be activated. They can transport the glucose. So, so this two transporters basically responsible for basal glucose uptake in both tissues. So the basal means that when particularly when blood glucose level not that very high, then this two transporters will not that activate, right? And GLU2 has a high Km or low affinity for glucose. That means only when you have very high blood glucose levels and GLU2 can be activated. For example, a good example, during and after eating, right? You raise the blood glucose level, so then GLU2 can be activated and take up a lot of glucose into the liver or into the pancreas for insulin secretion. And GLU4 is has the low affinity and high low Km, high affinity for glucose. And GLU4 is a little bit different from the other type of transporters because it responds to insulin, right? So even they have, so only when insulin level is high, and also GLU4 is translocated, right? When we don't have a high insulin level, majority of the glucose just stay inside of cell. And only when we have high insulin and then can stimulate GLU4 translocation to the cell surface. So again, GLU4 is mainly responsible for insulin-stimulated glucose uptake. Right? Okay. So lastly, I think would you guys just try to remember the two different transporters that respond to different conditions. GLU2 it basically is regulated by high glucose level, right? During the fat condition, being active and take up glucose, and GLU4 is also takes glucose during the fat condition, but it's regulated by insulin. So different regulation, right? Okay, so regulation. Just try to remember the three key reaction in the glycolysis and four key reaction in gluconeogenesis, right? And this key reaction or enzymes in two different opposite the pathways, they are coordinately regulated. So for example, during the fasting conditions, we wanted to increase the gluconeogenesis, and we wanted to slow down the glycolytic pathway, right? Glucose oxidation pathway. So that means something that we need to regulate to raise the, so increase the enzyme activity in the gluconeogenesis and to suppress the enzyme that in the glycolytic pathways. So this is just a general, idea about the regulation, how these two pathways being coordinately regulated. And here I show you the substrate cycles and how glycolysis and gluconeogenesis are regulated coordinately. Again, for the gluconeogenesis on the left side is a glycolytic pathway, right? So from the top to the bottom, right, you oxidize glucose to pyruvate, and for the right side, from the bottom to the top, is the gluconeogenic pathways, right? And only listed the key reactions, 3 key reactions in glycolysis, and the 4 key reactions in the gluconeogenesis, and how these key enzymes in 2 different pathways are regulated by substrate and hormones. So I just wanted you to focusing on some kind of important substrate and hormones, right? For example, fructose 6 phosphate and fructose 1 phosphate, and citrate, AMP. These are very important. The substrate that can regulate activities of the key enzyme in glycolysis, right? And for hormones, I want you to focusing on the insulin and glucagon, epinephrine, right? And how they regulate enzyme activities in the 2 different pathways. I just want to take one example for insulin. So insulin is levels increase during the fat condition, right? So insulin should stimulate the glycolysis. So you can see the plus means the stimulate and minus means the inhibits, right? So insulin increases the enzyme of glucokinase in the glycolysis. This is the key enzyme, right? One of the key enzymes, so the plus. But it actually inhibits the gluconeogenesis, so minus, for this key enzyme, glucose 6-phosphatase key enzyme in gluconeogenesis, which makes sense, right? In the fat condition, we wanted to oxidize more glucose, and we don't want to make glucose, right? So insulin stimulates the key enzyme in the glycolysis and inhibits the gluconeogenesis key enzymes. And similar for the second pair of the the reaction, the key enzyme, like insulin stimulates the phosphofructose 1 kinase, but inhibits the fructose 1,6-biphosphatase in the gluconeogenesis. Same thing for the third pair of the reactions. And we will go to the more details later. Any questions so far? Okay, I think probably it's time for the quiz.